Welcome to the NPTEL course on Bioinformatics. I am Dr. Michael Gromicha from the Department of Biotechnology, IIT Madras. In this lecture, I will provide an overview on bioinformatics, different aspects of bioinformatics and the applications of bioinformatics in different complexities of biological systems. In subsequent lectures, I will describe the details on various aspects. In this course, I mainly follow the books Protein Bioinformatics written by me and published in 2010 by Elsevier Academic Press, and the book by Kren and Reimer, Fundamental Concepts of Bioinformatics, published in 2006. For the general concepts of bioinformatics, I will use the Kren and Reimer book, and for the applications on various aspects of proteins such as protein sequence analysis, protein structure analysis, protein structure prediction and protein folding, I use my book on protein bioinformatics. So, what is bioinformatics? If you split into two parts, bio plus informatics. So, applications of the informatics on biological systems. So, I put the bioinformatics in the central part. So, it is a field of science in which biology plays a major role, right, and combined with other applications from different fields of science such as computer science, information technology and others, to merge into a single discipline to analyze the biological data using statistical techniques as well as the computer algorithms. So, if you see this diagram, so I put the computer science, the bioinformatics at the middle with all other fields which are linked with bioinformatics. So, if you look into this breadth of bioinformatics, in fact, the analysis of biological systems have been carried out for the past several decades using a small scale analysis, and in 1979 Pauline Hergweg coined the word bioinformatics to deal with the applications of the different science in biological systems. So, if you look into the different fields of science, how the different fields contribute to the birth and the development of bioinformatics. For example, computer science, can you tell one example how the computer science contribute to bioinformatics?
Machine learning can be used probably, and in computer science there are a lot of computer algorithms are being used for solving the biological problem.
Correct. So, you can develop several programmings and you can extract the hidden data available in biological informations. Also, you can use different machine learning techniques and other algorithms to understand and to capture the information as well as for the prediction purposes. So, can I tell an example how the mathematics or statistics is used to the development of bioinformatics?
Example would be the use of statistics. For example, in the use of Ramachandran plot, we plot the phi and psi angles of protein backbone.
Correct. So, you can use mathematics, right, to derive some principles and to relate the data, for example, the protein sequences or how the distribution of the residues in the Ramachandran plot and so on, using this mathematics, and you verify the data whether you obtain any model. So, whether they are statistically significant or not. You can relate with the correlation analysis, you can relate the regression techniques, as well as you can see whether the data are statistically significant or not. So, now if you use the information technology, how the information technology contribute to the development of bioinformatics?
There has been an increased computational resource over the decades, which is used to do a large scale data analysis.
Correct. So, you can use for large scale analysis, you can develop the online resources, right, you can see the computer storage and so on. So, in this case that will enhance the applications of bioinformatics to various fields. Likewise, physics, if you talk about the physics, so you can see the concept of the various types of interactions like electrostatic interactions or van der Waals interactions or hydrophobic interactions, how these interactions are important to understand the folding mechanism of proteins. For example, if you take protein folding, in the unfolded state of a protein, so it is like wobbling, and you can see it is very like a random coil conformation. When this protein folds into specific 3-dimensional structures, it can form a specific 3D structures, and how a protein can attain a specific 3D structures from its sequence, right. This can be explained by various types of interactions like disulfide bonds, electrostatic interactions, van der Waals interactions. So, understand the principles governing the folding state of a protein, it requires the physical concepts. So, we can use physics to understand the mechanism of the folding of any globular proteins. So, if we consider all the fields, if you see life science, computing, maths, stat, information technology or physics or chemistry, which one is the major field for the birth of bioinformatics? life sciences, right, because we need the data. So, without data, even if you have several fields, we cannot apply it to different data, right. So, we need a specific data. So, data, where shall we get the data? So, you can get the data from biological experiments. So, if you look into these life sciences, there are produced lot of data, right, on various aspects such as the macromolecular sequences, for example, DNA sequence or protein sequence, right, and the structures, protein structures, DNA structures, complex structures and so on, different expression profiles, different pathways and so on. So, how the bioinformatics is used to understand the data? So, bioinformatics is help to acquire the data, to manage the data and to analyze the data and to understand the data, right. So, bioinformatics is the major field, right, to understand the concepts and understand the hidden information available by the experiments produced in life sciences. So, what are the various aspects, how the bioinformatics is grown, what are the various applications of bioinformatics, how the bioinformatics contribute to society, right, the various aspects. So, briefly I put into 5 bullet points. The first one is well organized databases. The biologists, they do the experiments and they produce data and they publish the data in the literature. It is very important to collect all the information and put in a form of database. For example, if data are available scatteredly here and there, right. So, it is very important to collect all the information and put in a proper form, right, and then you give some options to extract the data from this database. This is what the bioinformatics do to develop plenty of databases which are well organized and in computer readable form. Once we have the data, right, sufficient number of data, which is very essential and it is required for any analysis, right, otherwise you do not get any statistically significant data. So, second option is, once you have the database, you can derive hypotheses. What will happen? What is the relationship between some specific features as well as any function? If you know the function or if you know any specific characteristics of any biological systems, what makes these characteristics to a particular systems, right. So, here we try to develop some features, right, and using these features you can link the function of any biological system, right. For example, if you take a protein sequences or protein structures, there are different types of proteins, like for example, if you get a protein structures, globular proteins, membrane proteins and different types of globular proteins, say alpha proteins, beta proteins and so on, and whether we can identify this type of proteins from a pool of sequences, right. So, you can left side you can see the sequences, because if you have the sequence, you know the information regarding the amino acid residues, right. So, you can get the number of residues of each type. So, there are 20 different types of residues, right, present in these proteins, and you can relate this. If there is dominant with some specific residues, for example, hydrophobic residues, then you can say that this could be a transmembrane helical protein. Likewise, for any proteins having different functions or different type of diseases, right. So, you can derive some hypothesis what is the basic principle for having the some biological systems. Once we derive the hypothesis to understand, okay, these are the major factors for any specific systems, right. Then the next step is whether we are able to describe any algorithm, whether we are able to derive any algorithm, right. So, if you see the features and if you see the functions and if you have a relationship, so here you have the features this side, here you have the function, right, whether we can relate these features and function, then what is the mathematical equation to characterize the function in terms of features, right. So, you can do a function, right, to understand the function from the features. This, when we do this, we can make the algorithm. Once the algorithm is ready, then we can use it for public. In this case, we use web servers or online applications, right. With earlier days, when the internet was not fast enough, so at that time, see, everyone they create the servers, they create their own algorithms, they keep themselves. It is difficult to transfer, but currently due to the advancements of this computational biology and the computational techniques and fast internet facilities, several observers have been developed to give the applications to others, right. In our laboratory also we have developed various tools, right, which are widely used in the literature. So, many people use these databases as well as the tools, right, to understand any biological systems. So, the fourth one, so a little bit discuss about the virtual screening. So, for example, the case of drug design, currently it is very popular, because there are a lot of small molecules are available in the literature, and the people are affected with several types of diseases like the cardiovascular diseases, cancer and so on, and currently we are developed with the chikungunya, dengue and so on. So, in all these cases, to identify drugs, they try to find a target, and then we see what are the functions of the particular target, what are the actions, what are the important residues, right, and then we try to inhibit the activity so that we can reduce this disease. So, here is one example for the structure based drug design. So, if we have a protein, so this is a target. So, here I show a target of CS kinase, because the CS kinases are very important for several cellular activities, right. So, there are several kinases, one is the CS kinase, here this is very important for the colorectal cancer. So, this is an attractive target to define the inhibitor for the colorectal cancers. To do this, there are various options how to derive a particular ligand to be an inhibitor, right. So, it is a very large pool. So, how to derive it? So, in this case I will tell one example. So, finding a fish in a pond. So, if you see it is a pond here. So, can you see different fishes in the different ponds? So, if you want to catch a fish, where will you put your net? In the number 1, right, number 1 or 2 if you put, you will get fish. If you put your net number 4, you do not get anything, even if you spend much time you do not get anything, right. So, if some of you tell you that okay, you are trying to catch a fish for a long time, okay, so you try to use this one to put in particular side, then if you do it and if you get a fish, then you will be very happy, right, because you do not want to waste your time. So, this is the case. So, for any disease, so there are different compounds, for example, compound 1, compound 2, compound 3 and so on. If you take compound 1 and you try, this is failed, so it is not a drug, and take compound 2, this is also not a good drug, and then compound 3 is probably a drug, right. There are millions of compounds. If you look into the literature, right, so there are lot of compounds. For example, if you go to a zinc database, there are 35 million compounds, and in NMN database 2.2 million compounds, and the natural compounds in Chinese medicine 35,000 compounds. So, if you want to try one by one, right, when you try everything, then the patient will die at that time. Second case, if you try to use each compounds experimentally, it will take long time, it needs long manpower, and also it needs lot of money, right. So, in this case how to do it? So, among these 35 million compounds, if someone can reduce to 35,000 compounds, then the number of experiments will be reduced by 1000 times. So, if instead of these 2.2 million compounds, if we have 100 compounds or 1000 compounds, then you can reduce enormously the search option. How to do it? So, here is a solution bioinformatics can do it, because currently we have very fast computers and we have very good techniques. So, it can assist one to searching drug target and designing drug from many millions of compounds. And the second one is how to use hypothesis. Okay, here I show an example. So, here I have 5 known values, experimentally known. So, we take the example number 1, the point 1. So, rice 50%, wheat 25%, meat 10%, fruits 10% and vegetable 5%. Now, if you do this, so then we can see that this is not controlled. For example, take the food pattern and weight control and go over the second one. So, rice 30%, wheat 5%, meat 10%, fruits 30% and vegetable 25%. In this case also it is not controlled. And we go for the third one, rice 25%, wheat 10%, meat 10%, fruits 25% and vegetables 30%. Here is controlled. Likewise, I give 5 examples. So, now I have a test case. Okay, this is a test case. Right, to another one consumes 20% rice, 10% wheat, fruits 20%, meat 10%, vegetable 40%. Right. So, now the question is here is controlled or not? What is the answer? It is controlled, right? It is correct. So, why it is controlled? How do you know this is controlled? Can you tell one example?
The fifth one.
Vegetables are high.
Vegetables high means here also vegetables are 25%, but it is not controlled. But here the vegetables is less. So, we can derive some principles, you can derive some equations using statistics, right. So, show one example, right, we can say that its answer is controlled, you are right. Okay, so, you can see the right and wheat is less than 35% and meat is less than 15% and vegetables more than 30%. So, likewise you can derive several equations, right. You can properly study the initial data set, that is experimental data set, and from this experimental data set you can derive some equations, right, some conditions where this will fit and apply these conditions to the new set of data, and then you can see whether that is controlled or not. So, likewise bioinformatics can handle large amount of data and provide possible solutions. Okay, now I will explain a little bit more about the virtual screening of the compounds. Here I show one example how we use the virtual screening to understand the drug design. So, I show an example here, this is the protein, right, this is the CS kinase in, protein. So, there are different domains. So, you can see one domain left side, that is a domain SH3 domain, SH2 domain, and here this is the tyrosine kinase domain, and the here is a catalytic domain, and here is a phosphorylation site, the tyrosine 416, and this is the loop. The question is, okay, this is very important for the colorectal cancer, it is a target. So, it is important to identify a probable drug target for this particular CS kinase enzyme. So, this is one aspect. Here one side now we have the protein, right. So, we have the target TS kinase, and the other side, so how to design an inhibitor. So, they have the library of NML library, we have 2.2 million compounds. Among the 2.2 million compounds, how to choose the probable compounds which can be a lead compound for a drug. So, now if you do it for 2.2 million compounds, it takes long time, it takes lot of money, because each compound cost about 30 to 40,000 rupees, right. So, you have to do they spend time to do for all the 2.2 million compounds. So, how to do that? So, in this case we can derive some methodology. First you see whether the structure is known. If the structure is known, we can use the particular structure, right. If the structure is not known, then we need to model the structure, and then we have to check for the activation sites, where are the activation sites the ligand can bind, and see the pockets which are the binding sites, right. Here is the site. So, now you see 2.2 million compounds, you check the features of all the compounds and make some conditions to fit with this particular pocket, right. Then you can use a molecular weight, you can use a hydrogen bond donor or the acceptors, right, various options you take, and then you eliminate the compounds. Finally, you can use virtual screening like docking you can do with these compounds, and finally you derive some compounds. So, in 2004 the Tokyo Institute of Technology, they organized a competition to identify inhibitors for this particular target. We also contributed in that, right. We identified about 120 compounds, and they tested 50 compounds among the 120, right, and we showed that 4 compounds showed inhibition and 1 is the probable hit compound. They continued the same in the next year 2015, right. There are about 2000 compounds, right. They found 5 showed inhibition and 2 are hits. So, in the downside floor you can see this figure, I show the how the ligand interact with the protein. You can see the green ones, right. So, the green one shows the ligand and the surrounded ones are the protein side. So, these ligands they have some specific interactions. You can see the hydrogen bonds or the hydrophobic interactions and van der Waals interactions, and they, because of these interactions, they tightly bind with the protein and they act as an inhibitor for this particular kinase. In the later classes I will explain about the more details on the structure based drug design. So, till now we discussed few aspects of bioinformatics. So, what are the different aspects we discussed? Databases, the one is well organized databases, bioinformatics contribute to organized databases, right. So, then the second one, computer derived hypothesis, when you have the data, then you can develop several functions, right. So, to relate the features and the functions, right, and once we derive the hypothesis, then we can develop several algorithms, right, for the prediction, and then once we predict, then we can make it as online applications in the form of observers. There are several observers for example protein structure prediction, protein function prediction, right. So, how the DNA can bend and how the DNA can interact with the proteins and so on. Then the fourth one we discuss now regarding the virtual screening of compounds, how they do the screening for the drug development, and currently if you see the bioinformatics is widely applied in next generation sequence analysis. Now, all are interested in personalized medicine. Few years ago it was very expensive to sequence a genome, now it is very cheap to get a sequence, right. So, everyone wants to see their own genome and what are the proteins they have and what are the functions they will do, and to understand what is the probability of having any specific mutation to your protein and so on. So, in this case now currently we have a lot of data, right, from obtained from the next generation sequences, right, for example the Illumina sequencing, right. So, because due to the advancements in the sequencing techniques, now there are a lot of data available in the literature, and the question is how to analyze the data. So, how to extract information from these specific sequences, right. So, there are several ways to get the sequences. They have use a short reads and to get the final sequence. For example, if you find some patients which are affected with the cancer, affected with the Parkinson disease and the Alzheimer's disease and so on, how the genome or proteome is different from healthy individuals. So, they get the data for the patients, right, and they get the full sequence, and they get the data from the individuals, healthy individuals, and they compare. So, how are the features, what are the variations, what are the mutations, right, whether the mutations are in the protein coding regions or non coding regions, and then they relate how these mutations or these residues are important, where they are involved in different pathways and how they are influencing the different diseases. They try to see the information and then they go with the treatment. For example, if you are affected with cancer or any specific diseases, they are treated. So, they go to the hospital, they get the patient's data as well as they get the information regarding drug and the drug response, and they make a database, we can do it, right. So, from that information you can see that if any specific variations, okay, this specific drug will work. So, if we have this information, then this will be helpful for the personalized medicine for different diseases. Likewise, bioinformatics plays a major role on different aspects in human health as well as for medicine.